This is a GCSE Biology presentation looking at limited factors and the AQA higher only sections. To get started, describe and explain examples of factors that can affect the rate of photosynthesis. There are some pictures there to help you. Pause the presentation, come up with your answer, and restart the presentation whenever you're ready to check your work. Make sure you describe and explain to get the maximum number of marks. In terms of describing, remember sunlight intensity, we can't just write sunlight. We have carbon dioxide concentration, again carbon dioxide on its own is not enough. We have the amount of chlorophyll and we have the temperature. And in terms of explanation, you need to be able to link each of those different properties to the rate of photosynthesis, and remember temperature is especially detailed because you need to bring in the concept of high temperatures and denaturing. There is a separate presentation looking at factors affecting the rate of photosynthesis. If you're not sure, please Google and check that one out. This presentation is only looking at the higher content for the biology course, so if you are not studying the higher paper, then do not go any further in this presentation. Now this may seem a little odd at the start, but you'll have to trust me on this. This is one of the easiest ways to pick up the concept of limiting factors. So what do you need to make a fast food Happy Meal? I'm not going to advertise any particular version of a Happy Meal or any particular fast food restaurant, but I'm sure you can work it out. So what do you need? Well, you need four things. You need a drink, a burger, a toy, and a set of fries. There's lots of variations I know, but this is the bog standard one we're going to work with. If we have an example of where someone working at McDonald's is responsible for making the Happy Meals. If they have 50 drinks, 50 burgers, 50 toys, and 50 sets of fries, how many Happy Meals can they make? Well, the answer is 50. If, however, something's happened and they've only got 40 toys, how many Happy Meals can they make? Well, the answer again is 40. The reason it's 40 is because that even though they have 50 drinks, 50 burgers, and 50 fries, once they get to number 40, they no longer have enough toys to make a complete set. If we go again, and this time say we've only 20 burgers, the maximum number of Happy Meals we can make is 20. Because we're limited by the number of burgers, it doesn't matter how many drinks, toys, and fries we've got, we will only make a maximum of 20 meals. Notice that the burgers in this example is the one on the lowest amount, and it would be called the limiting factor because it's limiting the number of Happy Meals we can make. In the previous example, it would have been the 40 toys that would be the limiting factor because it is the variable in the lowest amount and is limiting the number of Happy Meals. If we increase the number of burgers up to 41, then the number of burgers at 41 is no longer the limiting factor. It's the toy that becomes the limiting factor because there's only 40 of them, and they of course are determining the rate of Happy Meals and setting that at 40. Earlier I said you'd have to trust me in terms of what's this got to do with limiting factors in photosynthesis. Well, if we replace the drink, the burger, the toy, the fry, and the Happy Meal with light intensity, carbon dioxide concentration, amount of chlorophyll, and the temperature, and the number of Happy Meals becomes the rate of photosynthesis, all the same rules apply. If we were to do a graph for the number of toys and number of Happy Meals, as we increase the number of toys from 0, where we've 0 toys, we've 0 Happy Meals, we would increase the number of Happy Meals. Eventually, however, we would get to a point where even though we had more and more toys, we had run out of one of the other things that makes up the Happy Meal. And as a result, the graph would level off. Remember, do not use the term plateau. In biology, we only use the term level off. So all you have to do is describe the graph. Remember, when you hear the word describe, it means what. So what is the graph? What do you see on the graph? Pause the presentation, and when you're ready, you can restart. Pause now. So when we're describing the graph, there are two separate sections to the blue line on the graph. There's part A to B and part B to C. So we talk about them separately. And we simply talk about as we increase factor X between points A and B, we increase the rate. However, when we increase factor X between points B and C, the rate remains constant. Now, we haven't done any explanation as to why that's happening because the question is asking us to describe, which means what is happening. In this question, we're being asked to explain the graph. So again, pause the presentation, come up with the best answer you can, and when you're ready, you can restart the presentation. Pause now. So once again, we'd use the different lines in terms of A, B, and C, but this time we'd say that between A and B, increasing factor X increases the rate, and the reason it does that is because we have factor X as a limiting factor. We know it's a limiting factor because when we increase it, the rate increases. However, between B and C, there is no increase in rate, even though we're increasing factor X. And that's because factor X is no longer the limiting factor between B and C. Hence, increasing it doesn't increase the rate. Notice the use of the word because. When you're explaining anything, you always should have that word because in there. Why is something happening? This is a more common example of what you'd find on a higher exam. In this particular thing, what we've got is two separate lines on the same graph. We've got the line A, B, C, which is the blue line, and the line A, B, D, E, which is the blue/pink line. Remember, between A and B, the pink line still exists. It's just hidden behind the blue that we can't see it. Well, there's clearly something else going on, and we clearly have to have a difference between B, D, E, and B and C. The blue and pink, something's different. And what it will be in the exam is that there will be some other factor related to photosynthesis that will be different for E and different for C. So for example, if on the bottom of the graph on the x-axis we said there was carbon dioxide concentration, then the difference between E and C could be different light intensities, could be different temperatures. But there will always be a separate factor from the factor at the bottom of the graph and the factor that separates the lines on the graph. Remember, when the line is horizontal, whatever that variable is not a limiting factor. When the line increases, then we know increasing that particular variable is increasing the rate of reaction, and therefore it was the limiting factor. So in this example, we've now put four separate lines into one graph. We've identified light intensity as one of the things we're changing, carbon dioxide concentration, and temperature, all in the same graph. If you're given a graph like this in the exam, my strongest recommendation is that in the exam you actually put some annotations to the graph so that you can identify the different variations. So for example, the difference between the pink and the blue line at the top is the fact that the blue line is at 15 degrees Celsius, whereas the pink line is at 25 degrees Celsius. So there I would identify that the temperature is the difference between the two. If we look at the graphs lines at the bottom, the orange and the green, they both have the same concentration of carbon dioxide, but again, the temperatures are different. And then lastly, I can compare the lines at the bottom with a concentration of carbon dioxide at 0.04% to the concentration of carbon dioxide in the top two lines, which is 4%. And the differences between the temperature and the differences in the concentration of carbon dioxide explain why those graphs go up in different directions.
Remember, at point A, again, we have zero light intensity. So if we have zero light intensity, we have no photosynthesis. As we increase the light intensity, the orange and the green line are limited, but they're limited by temperature, which is why the two different lines, one's higher than the other, the orange is higher up. But they're also limited by carbon dioxide concentration. And no matter how much you increase the light intensity or the temperature, until you increase the carbon dioxide concentration to higher than 0.04%, that line will remain horizontal in both the orange and the green. It also explains why the blue and the pink lines go much higher. Because they have the higher carbon dioxide concentration, the time that it takes for that particular variable to become limiting, we have to increase light intensity quite a bit. So the point where the blue and the pink line meet is where the light intensity is no longer the limiting factor. When the blue line becomes horizontal, light intensity is not a limiting factor, just like we had at the very first set of graphs. What is becoming a limiting factor is temperature. And the reason we know this is because if you take the blue line and you increase the temperature from 15 degrees to 25 degrees, you can see we get the pink line where it continues to go up. The rate of photosynthesis continues to go up, but again will level off. We don't know when pink line levels off, if the limiting factor is the carbon dioxide level or if the limiting factor is the temperature. But we do know between the pink horizontal line and the blue horizontal line that temperature is the limiting factor, because if it wasn't, then increasing from 15 degrees Celsius to 25 degrees Celsius would not result in any change in the rate of photosynthesis. The fact that increasing the temperature does change the rate of photosynthesis indicates that the temperature is the limiting factor. Now we have this thing called a light compensation point, and a light compensation point is when we're looking at photosynthesis and respiration at the same time. And what it essentially is, it's the light intensity that anything that carries out photosynthesis gets exposed to. And it's that light intensity, that the rate of photosynthesis and the rate of respiration result in no net gas exchange in and out of the plant. What that means is that in photosynthesis, CO2 is used. In respiration, CO2 is produced. At the compensation point, the rate at which the plant is producing CO2 in the process of respiration, it is using that CO2 in exactly the same rate in photosynthesis. So there is no CO2 given off and there's no CO2 taken up. And that's referred to as the compensation point. This last bit in the higher paper relates to the common practical that's carried out in photosynthesis, where you get some kind of plant in water and you move a light source closer or further away from it, and you measure the rate of gas production by that plant in the water, either in the form of bubbles or in the form of volume of gas given off, and you can make an inference in terms of the rate of photosynthesis. Well, when we move the light intensity, you automatically assume that if we increase the distance from the photosynthesizing organism, the further away the light is, the lower the light intensity, and therefore the slower the rate of photosynthesis. And again, if you move the light source closer, you would increase the rate of photosynthesis. However, there's actually a relationship that we can mathematically show in terms of the change in distance and the change in the rate of photosynthesis. The relationship is described as inverse square law. Now, what that means is that if we had a thing called square law, that would mean the light intensity is proportional to the distance squared. Inverse square law simply means that light intensity is proportional to 1 divided by the distance squared. And the idea is that as we move a light source further and further away from the photosynthesizing organism, the light from the light source has to cover a bigger area. It has to cover a bigger area, not just a direct straight pathway. And if you look in the diagram on the left-hand side, you get the idea of what I mean by double the area. So when we actually move a particular distance, we actually have 1/4 of the light intensity, which means that doubling the distance actually results in a quartering of the rate of photosynthesis. And if you put that into the equation, well, D is 2 because we've doubled the distance. 2 squared is 4. So 1 divided by 4 means that our rate of photosynthesis has decreased by 1/4 if we double the distance by moving the light source further back. By the same token, if we move the light source closer, we would actually increase the rate of photosynthesis by a factor of 4. Now, the inverse square law, you don't need to go into a huge amount of detail about why it happens. You just need to know the relationship between the light intensity and the distance that you move away or closer to the photosynthesizing organism. You can express it mathematically, 1 divided by distance squared, or you can simply talk about the fact that it's inverse square law. But it comes up very, very common on the exam, so make sure you do wrap your head around the basics. Remember, it's proportional, not equal. The numbers are never equal, it's just the amount of change in one results in the same amount of change in the other. So if we double the distance, then we are doing 1 over the distance squared in terms of light intensity. Now, just to finish off, all you have to do is a learning triangle. Remember, we're looking for 3 things you knew before the lesson started that you can remember that have come up during the lesson. We're looking for 2 things you've learned this lesson, and 1 thing you're still not sure about, or 1 question to test yourself on, or 1 thing to go and find out, something that you're going to use for review purposes.